Hello students, good morning. Welcome back to a new video of Sunanda's tutorial. In this video, we will discuss about pBR 322 or it is a plasmid cloning vector pBR 322. So pBR 322 is an example of plasmid vector. Okay. So pBR 322 or plasmid BR 322 is a plasmid cloning vector. It is widely used in the field of genetic engineering and in biotechnological studies. So at first it was developed in 1977, in the year 1977 by Francisco Bolivar and Raymond Rodriguez. So according to their name, the initials of their name, this plasmid is named as plasmid BR and 322 is just a numerical designation. Okay, it is just a number which distinguishes it from other plasmids created in their lab. Okay, so at first this plasmid is a circular double-stranded DNA molecule that contains gene for ampicillin and tetracycline. Ampicillin and tetracycline are two antibiotic resistance genes, and this plasmid also have origin of replication. So presence of these peculiar structure makes this plasmid as a fundamental tool for cloning and studies of recombinant DNA technology. Okay. Next, what are the key features of this pBR 322? At first, it is small, having 4361 base pair. So this plasmid used to carry foreign DNA into host cell like E. coli. Next, this plasmid have antibiotic resistance gene like ampicillin and tetracycline resistance genes. Next, this plasmid have restriction sites. So, inserting foreign DNA into one of these sites can disrupt the resistant gene, which is a technique called as insertional inactivation. So, you can insert a specific desired piece of DNA in that particular restriction site. So when you insert a piece of DNA in that particular part, that particular part gets inactivated, which is called as insertional inactivation, which helps to distinguish the recombinant and non-recombinant plasmids. Okay. Next is ORI sites. This sequence allows the plasmid to be replicated within the host cell, and this site is called as origin of replication. As these plasmids are self-replicative in nature, they can replicate by itself. So replication starts from a particular site called as origin of replication. Next, synthesis. It was one of the first artificial plasmids created and is fully sequenced. Okay, so this plasmid have 4361 base pair. Okay. Next, what are the advantages of this plasmid pBR 322? Due to its manageable size or small size, it is widely used as a cloning vector. Next, the presence of two antibiotic resistant genes eases the selection process of recombinants. Next, multiple restriction enzyme sites make the plasmid compatible in many ways. Next, it has a high copy number, which is highly favorable in genetic engineering studies. Then the disadvantages. At first, detection of recombinant cells having newly inserted DNA molecule is time consuming. So this is the disadvantage of pBR 322. Okay. So it is all about your pBR 322. It is an example of plasmid vector. This is the structure of pBR 322 or plasmid BR 322. Okay, so I hope you enjoyed this session, you enjoyed this lecture video. If you liked it, please hit the like button. Please share with your other friends and like my videos. Subscribe to my channel Sunanda's Tutorial. Thank you.